Researchers at Linköping University have uncovered some interesting information on how we hear. Welcome, my name is Emily. I created Definite Hearing as a way to increase awareness for the deaf and hard of hearing. So in today's video, we're going to talk about how we hear. Now, the sad truth is, I wish I heard this way. I have a sensory neural hearing loss. This means that there is something wrong with the inner ear cells of my brain and the auditory neural connection that really makes sound significantly quiet for me compared to a normal hearing person. Thankfully, I wear hearing aids, and they tend to boost the volumes and frequencies that I need. Hearing aids enable me to hear and understand sound and communicate effectively. I love my hearing aids so much. But that's not why we're here today. Today we're going to talk about these researchers' finding at Linköping University in Sweden. So let's start with hearing. What is hearing? Hearing is sound waves entering our ear, getting processed from waves to nerve pulses and sent to the brain. Sound waves come in lots of different frequencies, wavelengths, and things like that. This results in different pitches and intonations and sounds. It is up to the ear to translate that sound from waves into neural pulses so that our brain can understand and interpret the sound. In order for me to effectively explain what these researchers found, I have unleashed my drawing skills. This might be like super bad, but it's the best I can do. So here we go. This is the ear. This is the middle ear where we have the incus, malleus, and stapes bones. These bones are the smallest in your body, and guess what? You can fit them in the size of your pinky nail. I don't know how that translates for people that have big pinky nails, but that's pretty small and pretty fascinating. So when the sound comes in, the malleus, incus, and stapes vibrate on the eardrum and send that wave to the cochlea. The cochlea is what we're focusing on today. The cochlea is where the action happens that the researchers recently found new information on. Within the cochlea, there are lots of little cells. The cells that we're specifically going to be talking about are the outer hair cells and the inner hair cells. When the sound comes in, these little cells are in charge of translating this sound from waves to neural pulses. The way that they do that is by calcium ion transportation. The researchers wanted to figure out more information on this aspect. Where are the calcium ions stored? How do these cells touch and communicate? And how is that sent to the brain? These are all excellent questions. It was long understood that there was a gap between a membrane right here, the tectorial membrane, and the inner and outer hair cells. The researchers found that this tectorial membrane, it was always hard to work with because it was clear, translucent. When they took it out of the ear, it would shrink, and it was difficult to work with. They found that this tectorial membrane actually reflected green light. So they were able to shine green light on it and understand it better. They found that, contrary to popular belief, there was not a gap between these inner and outer ear cells and the tectorial membrane. These cells use something called stereocilia, little arms and fingers, to attach to the tectorial membrane, as you can see here, and transfer the calcium ions that convert the sound from waves to neural synapses. This calcium gives the chemical reaction or the balance, the energy that needs to happen to do that. These cells then talk with the nerves that go to the brain. So in my sensory neural hearing loss, there is something wrong with this inner cochlea system. It's either something wrong with the nerves, maybe it's bad wiring, maybe the stereocilia don't touch the tectorial membrane and allow calcium to freely flow and that reaction to happen to translate the sound. We're not really sure. So there is a drug testing going on right now called FX322 that supposedly can rejuvenate these inner ear cells and therefore reduce the issues that a sensory neural hearing loss has. So if these cells could be rejuvenated, and if that was indeed the issue for my loss and not the bad wiring on the nerve, then my hearing loss could essentially be healed. How cool is that? Science is awesome. So awesome. The researchers at Linköping University were really excited to find that the stereocilia in the inner and outer ear cells actually embedded and touched the tectorial membrane. They found that the tectorial membrane was more of a reserve for calcium ions than just a membrane or a cover. It actually has a purpose according to their research. This is exciting. This is exciting for me because as a person with sensory neural hearing loss, I thought, okay, what if maybe my hearing loss could be influenced or positively affected if I drank more calcium, had more calcium in my diet? As a child, I was notorious for not having a lot of calcium. I hated milk. I was actually allergic to milk for the longest time. And so I kind of wonder, like, is there something there with FX322? If they rejuvenate those ear cells and then someone has a high calcium diet, could that help rejuvenate their inner ear cells? I have no idea. This is just me theorizing here. I am not an expert. I'm not a scientist. I'm not a doctor. I'm not an audiologist. I am not a professional. This is just me as a hard of hearing person telling you about this amazing research that's been done and how this study got me thinking. So what do you think about this recent finding? How does this finding of the stereocilia of the outer hair cells and the inner hair cells touching the tectorial membrane, aka the reserve of calcium, those ions that allow the transfer from the sound wavelength to the neural pulse, how does that impact your thought? What do you think? Do you have any thoughts on this? If so, I would love to hear in the comments below. The next step of this research is that researchers are going to try to understand how the calcium ions transfer from the tectorial membrane to the ear cells, the outer ear cells to the hair cells, the outer hair cells and the inner hair cells. So I'm excited to see that. I'm excited to see if they think of something new or come up with a new treatment. This is just exciting that humanity has had ears forever and we are still discovering things about how they work. And the more we figure out about how our ears work, the more that we can explore new options of treatment and care. I hope you enjoyed this video. Please like this video and subscribe to my channel. I look forward to making more awesome videos for you. Have a great day. Bye.